I'm gonna talk with the sea urchin populations in the Mediterranean because we studied this sea urchin population, and I want to show that it's different. We have to study both different spatial and temporal scales because we can conclude different things if we are studying just with just experiment, a few time experiments, and comparing the long-term series of data. So we have to know all these parameters for having a good knowledge about the dynamics of sea urchins. Well, first of all, I want to defend the Mediterranean, my love Mediterranean, because I want to say that it's a very diverse sea. We can consider it a hotspot of biodiversity. It has more than 17,000 described species inside. It's a very small sea, but it has about the 4 and 18% of the marine species. It means that the diversity in the Mediterranean is huge, and moreover, because his history of geologic and hydrodynamic history is it's a pump of endemics. So there is about the 25 to 30% of endemic species in the Mediterranean. So it's a very special place that we have to conserve and to understand for its conservation. And well, it can be considered an oligotrophic and semi-tropical sea. The productivity is very low. It's higher in the northwest Mediterranean, but in the rest of Mediterranean, it's very clean water, so the productivity is very low. And the temperature, here in the northwest Mediterranean, where I'm gonna talk about it varies between 12, 13 degrees in the winter, and between 24 and 26 degrees in the summer. So this is the mark. Well, for illustrating this high biodiversity, this Bouduresque and Barlac and these French algologists, they make a comparison with other seas, and well, just comparing the surface area and the number of the species, and the Mediterranean is one of the most rich seas in the world, with in this case with macrophyte biodiversity. So it's very diverse and it's very complex. But everything is small. I mean, we have not this big kelp, but there are small algae. But even with these small algae, they generate they make many other species that they can grow inside, and with the structure that they make. So, well, this is the framework we are working, and of course, this is a sea that there are about 21 states around the Mediterranean. There are many people around the Mediterranean, and of course, it's one of the most perturbated seas in the world. And the highest perturbation is the habitat degradation, because well, there's crowded, there is many people, exploitation, overfishing, and other type of exploitation, pollution, and it seems that climate change is one of the most actually perturbations that it's going to take part after the invasion, the species invasions, and other processes. So it's a very small, very rich, and very perturbated sea. So for we, the one of the most interesting things is to understand how is the processes and the factors that affect the communities, and why one rich algal communities with high biodiversity can become overgrazed communities dominated by sea urchins and with the poorest biodiversity. Because as Bob he told, these communities can stay in the same place at different times. So we can see in the same place 1 year this type of communities, and in other time we can see this completely different. And in this, in this argument, of course, the sea urchins are the key species that, that can create this difference. So there is this general model about the dynamics of the algal communities in the Mediterranean. Is made by Sala and collaborators in the 98, but they describe these two different states of the algal state, and these two states they has positive feedbacks that they maintain this state. So in the fleshy erect algae communities, because they're this is related to marine protected areas, but normally when there are many fish, these fish they prey on sea urchins and they maintain low sea urchins populations. When there is overfishing, the sea urchins population they grow and it becomes these sea urchin dominated communities with barren communities and low complexity. And these communities they have positive feedbacks, because if there is not predators, the recruitment is higher, so it is self-maintaining this community. And of course, if there is a prohibition of fishing or the predator fish, they come again, there is the possibility to come back to the prior community. Well, this is the general model, but when we go to the sea and when we are studying different places, this model works, but not at all. So there are some points that it works very good. For example, in some marine protected areas in Nevis Island, in Port Ross, there are few marine protected areas that this model works, but we can find many other different situations. So we can find sites where with fish, but many sea urchins, and we can dive in marine protected areas with many fish, but also we can see barrens in these marine protected areas, and also in places where there are no fish, we can see a huge communities of these Thistoseiras with this erect algae in perfect state. So the models works, but in a very general way. But when we are looking for specific places, we can find that there is some deviation in these models. I think this don't work anymore. Okay. Well, anyway. See? Okay, good. Well, so the thing is that the sea urchins is a key species in all this play, and well, for years, me and many other people in the Mediterranean, we dedicated our work to understand the dynamics of the populations of sea urchins. And basically, well, there are more or less the most important processes that regulate the sea urchin populations. And I will show that there are, well, I make a fast overview of small experiments to understand the dynamics of every one of these processes. But we'll see that these studies are at a very low spatial and temporal scale. And after that, we will see a long-term series of data of natural populations in the Mediterranean to see how these processes act in a natural populations. So the recruitment, we saw that the recruitment working with these traps of recruits. These are the small recruits we found. This is a series of 3 years with the sea temperature with the surface sea temperature, and we saw that the recruitment of sea urchins it's always during spring, late spring in March, June, July, every year. But this is kind of regular. The recruitment it occurs always here. There are other researchers that they found a small peak of recruitment in in the end of summer. But we saw that there is some regularity. Every year there is some kind of recruitment, but the number of recruits, it can vary a lot between one year and another. And also, the timing of the recruitment can be for some weeks before and after. But the interesting thing is that there is this regularity. Okay, there is a recruitment every year, but the potential of recruitment is very different. And it's very different, a bad year, and a good year, we can find many sea urchins, many recruits of sea urchins that they have the potential to destabilize the adult population. And another particularity is that it's regular in depth, so the recruitment is mainly in between 5 and 10 meters depth within the surface and 10 meters depth, but not deeper. But at a spatial scale, it's very irregular. So we can find the size of these circles. It's related to the abundance of sea urchins, and we can find a huge difference between one places and others, separated only in tens of meters. So it's very irregular, depending probably on the orography of the currents or etc. Because we didn't find any behavior in the larvae to recruit from one place to another. So the recruitment is hazardous at spatial scale. Well, about the predation, we saw a very general pattern. We compared the predation rates inside the marine protected area, the Medes Island marine protected areas where the fish abundance is very high, and comparing the predation in the non-protected Nervi coast. We saw that, for example, in barren communities with any refuge for the shelters, that inside the marine protected area, the predation was very high. After 5 minutes, the recruits we pushed were depleted. But in the non-protected area, the predation was really low. But moreover, we saw that the heterogeneity of the bottom is very important for the survival of these recruits, because in barren communities with any refuge for the recruits, the predation was very high. But when we did this tethering experiment in communities where there were algae or even crevices where the sea urchins could shelter, the predation was very low. Even was null if they just inside of the crevices. So another time we have a general pattern when there are many predators, the predation is very high, but the type of the bottom, the availability of shelter can modify this pattern. Moreover, we find a relationship between the size of the sea urchins after the this is in test diameter sizes, and the size of the fish, and also different predators along the growth, along the size of the sea urchins. For example, we began with 2 millimeters sea urchins, and the small labrids were the main predators. But when they are higher, especially after 1 centimeter in diameter, the sparids, mainly Diplodocus argus, were the main predators. So when they are very small, there are many other predators than Diplodocus argus that they can eat. Moreover, in recent studies, we find that these micropredators, these hermit crabs or shrimps or brachydians, etc., they can be very important predators of small sea urchins. And we find that this may be another positive feedback mechanism of maintaining these different states. For example, when we find in a forest state, there are this structural complexity of the algae, they allow many of invertebrates live inside of the canopy. So there are many abundance of predators, as we saw. And these predators, they eat these small recruits. So these small invertebrates, they maintain, they can stop this settlement. But in the barrens, we have not this abundance of these micropredators, and the settlers can grow and can be can have a higher survival than in the barren community. So it makes these positive feedbacks that can maintain these things. We saw also a behavior and an effect of the behavior. Well, basically we saw that looking for the movement of sea urchins inside and outside the marine protected areas with and without predators, we saw a different movement pattern. So we saw without predators, they move higher, more distance, especially during the night, that when there are fish. So it seems that the fish inhibit the movement of sea urchins. And this is important because doing more experiments with different situations with and without fish and sea urchins, we saw that this is an experiment of transplanting algae. We saw that the height or the biomass of different species of algae is affected differently by sea urchins or fish. So we saw that, well, this is the control algae that they grow. They were maintained during an experiment of 5 weeks. We saw that when there were sea urchins, these algae were predated, were eaten. Fish, herbivorous fish, also they eat some algae. But curiously, when there were fish and sea urchins, they feed less than when sea urchins were alone. So it means that there is an inhibition of sea urchins, of fish by sea urchins. Well, mainly this is that the general patterns we saw that the settlement has the potential for destabilizing the populations because they can arrive thousands of larvae in one determined place. The predation is a control process because the predators they can eat these sea urchins, but there are some processes, some contingencies that they can move, they can vary these general processes. So this spatial and temporal settlement variability, the refuges of the larvae, the sizes, the relationship with sizes with fish and sea urchins, the behavior, and we'll see the sea surface temperature that can modify this settlement, etc. So, and the special thing is that if you see, we were doing these experiments, and I show only my experiments here, but there is many people in the Mediterranean that they are doing this kind of experiments. But the thing is that these experiments are very short in time. I mean, in space, this was 3 small coves, and during 5 weeks. So with these experiments and the predation experiments, it was 20 minutes experiment. So with these small scale experiments, we are telling big histories. No? And the thing is that if we do a spatial and temporal scale plan, the thing is that all important processes occur here, or there are other things that we can know. For example, these ices or storms that occur not very frequent, but importantly, or even climate change that occur at very high scales. So this model, this general model, was made with these small scale experiments, but we need data from the field, from natural populations with a bigger scales to fully understand the dynamics of these communities. So, well, more or less, for asking this question, what is the important relative of these processes? Because, well, is the thing, if I do an experiment of predation, I can say, well, yes, the predation is the best, the highest process that that regulates sea urchins. No? And especially, well, is this the importance of the scales in understanding these things, and all the processes that can regulate these general processes that can modify the dynamics of these populations. Well, and for this, well, this was a small introduction, it was a kind of large, but what I want to show you is our series of data of different places that we study at trying to make the spatial and temporal scale bigger to understand the dynamics of the sea urchins population. So we have monitoring programs in Medes Island Marine Reserve, also in Port-Cros National Park that is in France, and we have also data from Scandola Marine Reserve that is also a marine reserve in Corsica that we monitor for 13 years by now. So this is a more or less long series of data, regular monitoring data that we'll see they can tell us some more things about this. Well, the Medes Island, well, you know, it's here in the northwest of Spain, in the Mediterranean, and it's a very small island. I mean, it's from here it's about 1 kilometer, so it's very small. It's very close to the coast, but there is a huge gradient of fish. So there are many fish here, and in the non-protected coast, well, there are also fish compared to other places in the Mediterranean, but not with the abundance of the Medes Island. So, well, and the monitoring programs is always the same, more or less, here in the Medes Island and in Port-Cros National Park and things. So we work in the same places with fixed transects, about 150 square meters, where we count all sea urchins and we measure the size of the skeleton. And we are comparing inside and outside the marine protected area. And also we compare two different structural habitats, these boulder bottoms, where the sea urchins can have a refuge and shelter from predators, and also bare rocks without these shelters, so they are exposed to the predators. Well, the big result is this. As Dr. Lesius showed us, we have some more ears here, but this is the general picture. So comparing, this is the non-reserve in boulder bottoms, and the marine protected area in boulder bottoms. And we see, first of all, that the habitat, the type of habitat, make a big difference between where in boulder bottoms there are more abundance of sea urchins than in vertical slope rocks. And another thing is that we didn't find any significant difference between inside and outside the marine protected area. So following the models, it is contradictory, but statistically we didn't find any difference between inside and outside the marine protected area. The only difference we found, it was these peaks of abundance that correspond to recruitment peaks. There are some years that the recruitment is very high, and outside the marine protected area, the abundance, they grow very, very fast. But after this peak abundance, the populations, it becomes lower because probably of mortality. The difference, the important difference is that these peaks of recruitment inside of the marine protected area it doesn't exist. So this is probably because of the predation pressure of the fish. So the predation is important for maintaining this, for controlling these recruitment peaks, but not at long term for making big difference between inside and outside the protected area. so the habitat is important, the fish predation is important, maintaining these habitats. Well, I told from here, well, here I was in a postdoc stay and I didn't make these controls, and after, I will talk later, there was a huge storm in all this coast, in the Mediterranean coast, and the sea urchins population, they go down very, very fast. I will talk about this now. So another thing we found is this relationship between adult sea urchins and recruits. We found inside the Medes Island marine protected areas where the fish predator abundance is very high, that was a very good correlation between adult and juveniles of sea urchins. So the adult sea urchins facilitate the survival of juveniles. But we didn't find this correlation outside of the marine protected area. So it means that where the predation abundance is very high, the predations make that the juveniles, well, depending on the adult population. And it means that the recruitment peaks, these high recruitment peaks, if there is not this relationship in the non-protected area, can make this growth of these populations. So it makes a mix between recruitment and mortality. So we find these peaks of recruitment, and looking the size structure of the population, we find that effectively it was a peak of recruitment. And I have to work a little more this data, and we find and I try to find a relationship with the sea surface temperature and the recruitment, and only see a certain relationship between the sea surface temperature on April and the recruitment. So in April, it is the time when the larvae are in the plankton, and probably they have a more survival with a higher temperature. Jose Carlos find and other people find this relationship. We find this relationship, but it's not very, very clear. And we had, and as I said, in 2008 we had a very huge storm in the Costa Brava, in this part of the Mediterranean coast, and this storm it was very important. I mean, it makes a huge wave, about more than 10 meters, and it's very unusual in this part of the Mediterranean, and this causes a very high impact on the benthic communities, because the impact it was mainly because of this huge wave. They move rocks on the bottom, and these rocks makes a high erosion of the bottom. So comparing before and after this storm, all the bottoms, boulder bottoms between 0 and maybe 15 meters depth were completely depleted. So were only the rock, and all the cover, algae, and of course sea urchins were crushed and depleted. So after this storm, we saw that, it caused a huge mortality, more than 80% in some places. And after this storm, the populations inside the marine protected area, in boulders and on vertical rocks, doesn't grow up very, very fast. But in the boulders of non-marine protected areas, they grow very, very high. This is because it was a huge recruitment peak after the storm. And this means, again, that the recruitment, this recruitment peak increased the populations inside the marine outside in the non-protected area, but not in the protected area. Why? Because in the marine protected area, the predators, they can control this recruitment peak. So again, there is this interaction between recruitment and predation that they control the population of sea urchins. And the important thing is that this storm caused a shift on the dynamics of the population. Well, about Arbacia lixula is the other most abundant species in the Mediterranean, but in this part of the Mediterranean, in the northwestern Mediterranean, is not very abundant, and we found also some regularity in the abundance of this species. We didn't find any difference between inside of the marine protected area and outside. Only a smooth difference between the boulders and the vertical and the slope rocks. And well, this is a little weird because nobody I think in the Mediterranean found a peak of recruitment of this species. Always the size structure of the population was with a dominance with this intermediate size, but we didn't find very clear peak recruitments of this species. So, and this species is interesting because they told that it's a thermophilic species, and in the southern part of the Mediterranean, it's very abundant. Maybe it's the most abundant species, more than Paracentrotus lividus, but here we didn't find any significant difference between the protection, the abundance of predators, but also on time. And well, it's also discussed the interaction, the competition between Paracentrotus and Arbacia, but we didn't found a clear relationship between these two species. So we didn't find any competition between these two species. So the general model of the general things that we can find about this monitoring data is that in the boulder bottoms we can find a high oscillation because of the recruitment, but these peaks of recruitment are controlled by the predation pressure of the predators. And this predation can be mediated by the availability of refuges because of it can explain the difference between bedrock and boulders, and also the abundance of adult individuals that can facilitate the survival of sea urchins. And well, we have as Dr. Lesios, they told us if we are studying just one piece of time of this area, we can have different conclusions about the dynamics of this population. But if we have the whole picture, we can find that there is this oscillation, and there is this event, these rare but important events that can change completely the dynamics of these populations. And after that, we really don't know what will happen. We think that probably in the Medes Island, where there are high abundance of predators, finally they can control the sea urchin population, and the sea urchin population will be low. And contrarily, in the non-protected areas, the sea urchin population, they will grow up another time. So these rare events can change not only the population dynamics, but these different states, the dynamic of the whole community in the benthic communities. Well, another example of a long-time series that they can tell us different things is in Portcross National Park. This is a very old national park. It's the first national park in the Mediterranean. It was created in the 76. And we follow the Kinoran, not only sea urchin, but the Kinoran population in different places, in different depths. And well, the big picture is this. So we have data from the 70s, from the late 70s. This data, it was made by the French people, by Joan Malin, Boudurès, Camberlac, and company. And after 1993, our team, we began to monitor these populations since 2003, that it was stopped, and well, since now. Now I'm trying to go again to see what happened with these populations, but we have only data from until 2003. So the thing is that in the 79, there was a high abundance of sea urchins until the 81, and in the 81 it was described a disease of the population of these of sea urchins. This disease was made by it was a bacterial, and well, it was followed by the Boudurès, Cazzolina, and some people, and they describe a high drop of the communities, mortalities in some places about the 80% of the adult sea urchins, and they told, well, in summer and shallow waters, especially, there was a drop on these communities. And we found that effectively these populations, they drop until density is very, very low. This series was interrupted in the 84, and we began again in the 93, and the populations, they don't reach the densities that they have in the late 70s. And after that, the densities, they drop down until a number, a very few abundance in the 2003. So we believe that this drop is caused by the predation of fish, that they are very abundant in Portcross. Well, the thing is that we have not a comparison in the non-protected area in this case, because we have only these controls inside of the Portcross, but well, this is the most probably explanation. So we have again one rare event that was this disease, and after that the population they go down and down. This is Paracentrotus lividus, and Arbacea lixula. It has a different trend. So before the mass mortality, it has a very low low numbers. Well, we have to be careful with the scale. So it's an order of magnitude of abundances of Paracentrotus lividus, and we saw that it was not affected by the mortality. And after 10 years, the densities go up, and after that they were maintained in these levels. So one explanation of this growing up of the Arbacea lixula is that because of the mortality of Paracentrotus lividus, they don't have this possible competition by Paracentrotus lividus. So, and effectively we find a certain relationship between Paracentrotus lividus and Arbacea lixula that it can be it can explain this growth and this competition between these two species. Okay, so this is the thing, and we have another example of more or less long series of data of the sea urchin population that is in Scandola, in a marine protected area. Is in Garganello. We have an experiment. We are controlling different types of communities. We are controlling these forest communities, well-developed forest communities. We are following barren communities. There are many barren communities inside of the marine protected area, so it's weird. And mixed populations, so mixed communities. So there are communities that they are in the process of changing. So we did an experiment. So we did an eradication, a punctual eradication at the beginning of the experiment. We we take off all the sea urchins in different places. And just doing this one time, it was maintained all the sea urchin population after that very low. So it means that just doing one eradication, getting out the sea urchins, it changed a shift on the communities. So this is in terms of density and biomass, and the algal cover after this eradication, they go up. So it seems that a crash, a huge change on the abundance of adult, it can make a shift on the community. And this shift is maintained during all the time. But in the natural plots, in the plots that we didn't manipulate, we saw that there was also changes on this community. So we saw that the sea urchins biomass and densities, it was going down and down every year, and also the biomass and the algal cover in this barren community. In the forest it was maintained, but in the barren community the algal cover began to increase until 2011, until this year, and now they are similar to to a forest. So this change, it was natural. So it wasn't caused by any perturbation, and it makes us to think that these different states are not so permanent, but there also a natural change, a natural dynamics on this type of communities. So it's possible that there are a change, a regular change on these communities. Well, about the temperature, we didn't saw a clear relationship between the increase of temperature on the sea urchin population, especially on Arbacea lixula. Arbacea lixula is told that it's a thermophilic species, and probably with the growth of the temperature, it's going up, but we didn't see a clear relationship with this. So these are a series of sea surface temperature, the annual mean sea surface temperature on Medes Island and in Port Cross Island, and we saw that there is a significant growth of the temperature, but the sea urchin population, they go down. So we didn't find this clear relationship. In Port Cross, we find not for Paracentrotus, because there was this drop, but for Arbacea, we find a small relationship, mainly due by this difference between the 84 and 93. But, well, still, we have not a clear relationship. I told this because there is a famous article by Buduresc and Francoeur and these people that they describe a very high increase of Paracentrotus lividus and other species in the Mediterranean, especially in Corsica, in Scandola, and they saw that in 10 years the abundance, they go up one order of magnitude up. And they say that this is because the increase of sea surface temperature. So we didn't find this clear relationship. So now we know that the Mediterranean waters are becoming warmer, but the information of these species, Arbacea lixula, we are not very sure. For example, in one of these missions in Scandola, we did a big census of the sea urchins populations in the wall, in the reserve, and we didn't find these high densities on Arbacea lixula. So we find densities that it was in the order of this. So with this, I want to tell that there are changes that are very important changes that can change the dynamics of the populations, but we have to be a little cautious, especially with the climate change, because there are also natural changes that it can be explained by this general pattern. So, well, I want to make a little cautious about this. As Dr. Lesio said, that we have to fight against the people that they say that there is not a climate change. We have to be a little cautious also to say, okay, there is climate change and there is huge changes in all the communities. So we need more evidences to describe exactly what is happening in the benthic communities. So we have to, as a conclusion, we have to integrate all spatial and temporal scales to fully understand the dynamics of the benthic communities. We need long-term monitoring programs. It's very important. As Dr. Lesio said, it's not, for the scientists, it's not very interesting to follow a population that it happens nothing, so just following 1 year and another, it doesn't make result in that you can publish this and after 15 years. So, but it's important, it's very important to have this long-term series, because now, after many years, we can talk about all these things. So, and it's important to have long-term series data, because this exceptional perturbation can determine the dynamics of this population and the whole community. And if we are studying a short periods of time, we cannot find this the effects of these exceptional perturbations. And well, and about the climate change, can act on sea urchins population through different processes by facilitating the survival of larvae or many different processes. But I think we need still in the Mediterranean more evidences to determine in what kind of processes. So, well, thank you very much. And of course, I want to say that this is not only my job. There is many people here that did work on this data, especially in the Department of Ecology and other departments. So if I have to put all the names, it will be this screen full of names. But well, I want to thank to all these people about this.